Today we're going to talk about lipids, one of the four major biomolecules, and one of my favorite because it's the yummiest. Lipids are in the category as one of our other major large biomolecules. But unlike carbohydrates, nucleic acids, and proteins, they do not form polymers. They're all hydrophobic and have little or no affinity for water. That's because they consist mostly of hydrocarbons, which form non-polar covalent bonds between carbon and hydrogen, and have little oxygen. And the four types of lipids that we're going to discuss today are fats or oils, phospholipids, steroids, and waxes. Fats are used as storage molecules in organisms, and that's because they're very energy dense. In fact, fats contain about 9 kilocalories per gram in their molecules compared to proteins and carbohydrates that only contain about 4 kilocalories per gram. Before we go on and talk more about fats, I want to tell you a little story. About 20 years ago, I took a bicycle trip across country from Portland, Oregon to Portland, Maine. And while I rode across country, I tried to keep the weight on my bicycle as light as possible. So which of these foods do you think I ate? So you might think I was healthy and I was trying to do the best I could to eat the healthiest food, but I was really just trying to keep the weight down to the minimum. And so I picked the food that I also enjoy to eat the most, which I hopefully you got from the hints. I ate donuts. Donuts have a lot of fats and therefore they're very energy dense. They're probably not the healthiest food, but I was riding so much at the time it didn't matter. Okay, fats also are very hydrophobic, and they separate from water because water molecules form hydrogen bonds with each other and exclude the fats. Fats are constructed of two types of smaller molecules, glycerol and fatty acid. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each of the carbons, or an OH group attached to each of the carbons. Fatty acids are long hydrocarbons with a carboxyl group, or it's also known as a carboxylic acid, a C double-bound oxygen, and a COH. The fatty acid joins with the glycerol through a reaction where water is produced. Do you remember the name for that reaction? Yes, this is a dehydration reaction. Most fats are actually composed of 3 fatty acids, and so they're known as a triacylglycerol or triglyceride. When the carboxylic acid from the fatty acid joins up with the hydroxyl group from the glycerol, it forms this ester functional group. So we call the covalent bond that links these two an ester linkage. Fatty acids are what determine the structure of fats. Fatty acids vary in length and in the number and location of double bonds. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds. They're saturated with hydrogen. Unsaturated fatty acids have one or more double bonds between carbons. That means they have less hydrogen. And polyunsaturated just means that they have 2 or more double bonds between carbons. So again, these are fatty acids. They have long hydrocarbon chains with this carboxyl group. The difference between a saturated fatty acid and an unsaturated fatty acid is in a saturated fatty acid, it has a maximum number of hydrogens around it. In an unsaturated fatty acid, there is a double bond. Since carbon can only form 4 bonds around it, that means it's going to have less hydrogen surrounding it. Be careful, some people recognize that carboxylic acid, and they see that double bond there and they think that that makes it unsaturated. Remember, all fatty acids have a carboxylic acid. The difference is, in the unsaturated fatty acid, it has this double bond. Saturated fats form from saturated fatty acids. We find foods high in saturated fats come from animals. Saturated fatty acids are very linear in shape. That allows saturated fatty acids to stack up next to each other very close, and means that they're going to have high intermolecular forces between them, which means that their melting point is higher too. So we tend to see foods high in saturated fats, the fat actually stays solid at room temperature. Diets high in saturated fats have been shown to contribute to cardiovascular disease through plaque deposits. Plaque deposits are where fat actually builds up on the arterial wall. This can lead to a restriction of blood flow and can cause things like heart attacks, for instance, where you're not getting blood flowing to the heart or part of the heart. The plaque deposits also lead to hardening of the arteries, meaning that the artery is less flexible, and that could eventually cause the artery to shred or tear. Plaque building up in an artery is known as atherosclerosis. A general term you may also hear relating to hardening of the artery is known as arteriosclerosis. Another type of fat is unsaturated fat. Unsaturated fats have unsaturated fatty acids. Because unsaturated fatty acids have a double bond, that usually leads to a bent structure, which means that unsaturated fatty acids can't build up on top of each other. They don't have as strong of intermolecular forces between them, and so they have a lower melting point, and they are liquid usually at room temperature. Fats that are liquid at room temperature are known as oils. And we find foods that have a higher amount of unsaturated fats are found in plants and fish. Transunsaturated fat are another type of unsaturated fats where there's a difference around the double bond. In a normal cis unsaturated fat, the hydrogens are on the same side, and that leads to this bent structure and the properties that we see of normal unsaturated fats, like they're liquid at room temperature. Transunsaturated fats have hydrogens on opposite sides. This leads actually to a very linear structure similar to saturated fats. Now, trans fats are found naturally in some foods like butter, but they were produced a lot in manufacturing of foods through a process called partial hydrogenation. Full hydrogenation is where you actually add hydrogen into a cis unsaturated fat and make it a saturated fat. Partial hydrogenation is where you interrupt that process. And by interrupting that process, you can get some of these trans fats. And it gives properties that were very desirable in making some foods. For instance, they're semi-solid at room temperature, which means that you can make really good like frostings that have a semi-solid texture to it. They're less likely to spoil. That means actually you could keep and reuse oil that you used for frying more times than you would be able to do it if you were using a cis unsaturated fat oil. However, diets high in transunsaturated fats have been associated with a higher risk of cardiovascular disease. There's actually more likely to be a buildup of plaque inside our arterial walls if we actually consume more transunsaturated fats than saturated fats. There's also been other links to other health problems like cancer, diabetes, obesity, and more. Since there has been a lot of research showing a link between transunsaturated fat and these many health concerns, the FDA banned trans fat in 2018, or at least they banned large amounts of trans fat. You can still have less than 0.5 grams per serving. Okay, before we move on, let's do a little review of fatty acids. Which of these fatty acids is most likely to lead to atherosclerosis? Order these from most solid to least solid. Which of these does not describe a cis unsaturated fat? The next type of lipid are phospholipids. Phospholipids are super important in our cells because they make up the cell membrane of all living cells. They also make up internal membranes of eukaryotic cells, like the nuclear membrane, called the nuclear envelope. They're composed of two major parts, a hydrophilic head and hydrophobic tails. Phospholipids make up the membrane of all of our cells in a bilayer arrangement. That means there are phospholipids opposing each other, with the hydrophobic tails facing each other, and the hydrophilic heads facing the water in the exterior environment and the water inside the cytosol or inside the cell. This provides a barrier so that molecules cannot just pass through and enter in or out of the cell without some control by the cell. The hydrophobic parts or hydrophobic tails of phospholipids are made up of fatty acids. The hydrophilic head is made up of the glycerol and a phosphate group attached to that glycerol. So normally in a fat, for instance, there were 3 fatty acids attached to a glycerol. In a phospholipid, instead of one of those fatty acids, there is a phosphate attached. In addition, there can be other groups attached that also make it more hydrophilic, like this is a choline up here, a charged nitrogen group. Phospholipids, when added to water, will self-assemble, where they'll either self-assemble into a bilayer, like in both this bilayer sheet or a liposome, with the fatty acid tails facing away from the water, or they'll form a mycel, where all the fatty acid tails face each other and the hydrophilic heads face towards the water. Phospholipids are great emulsifying agents. An emulsifying agent is a substance which is soluble in both oil and water and allows oil and water to mix. Lecithin is a phospholipid found in egg yolk and soybeans. And lecithin is used to make an emulsion like mayonnaise. Mayonnaise is just basically an emulsion of oil and water. Steroids are another type of lipid that are characterized by 4 fused rings, and they can have attachments outside of those, like this attachment over here on cholesterol, but the 4 fused rings are what you look for to identify a steroid. Some steroids or sterols are hormones. A hormone is a long-distance signaling molecule. It's produced in one part of the body and affects another part of the body. Some steroids that are hormones include sex hormones like testosterone and estrogen. Testosterone, for instance, is a hormone that causes some of the characteristics that we see in males. Like in this peacock, we see these bright coloration feathers that we don't see in the peahen because of the production of testosterone that actually turns on this coloration. Another important sterol is cholesterol, which is found in animal cell membranes. Cholesterol is a non-essential sterol. Our bodies actually make it ourselves. And it's found in our cell membranes and helps maintain the fluidity, which means that when temperatures go down, it stays more liquid, and when temperatures go up, it doesn't get too fluid and break apart. Cholesterol is also an important precursor to a lot of hormones. You can see that this diagram actually shows cholesterol is used to make like hormones like testosterone and estrogen or estradiol. Since cholesterol is non-essential, consuming a lot of it actually can also contribute to cardiovascular disease. You may have heard of bad and good cholesterol. Well, the description of bad and good cholesterol actually relates to these other molecules known as lipoproteins. Lipoproteins are these big, huge molecules surrounded by phospholipids and containing proteins that can carry things like triacylglycerols or triacylglyceride and cholesterol throughout our circulatory system. And there are 2 major types of lipoproteins, LDLs, which is often referred to as bad cholesterol. LDL stands for low density lipoprotein. That means that they're higher in actual lipids than proteins. Lipids are lower density than proteins are. And LDLs actually tend to transport fats and cholesterol to arterial walls and lead to that buildup of plaque in arterial walls. The good cholesterol is actually HDLs, or high-density lipoproteins. High-density lipoproteins contain more proteins than lipids. HDLs are actually associated with moving cholesterol out of the blood system and decreasing plaque deposits. The last type of lipid is known as waxes. Waxes are a fatty acid chain connected to a long chain alcohol, and they undergo a dehydration reaction again, forming an ester linkage. You can see that ester linkage up here. Waxes repel water, they have a high melting point, and they're resistant to degradation. Waxes have a lot of uses by different organisms, including humans. Bees use wax to actually produce their hive structure, and it stays solid even when temperatures get warm. Plants release wax on the outside of their leaves to actually prevent the loss of water through evaporation or transpiration. And humans use waxes for a variety of reasons, including to protect surfaces from degradation, and we use them to make candles. So that's all I have for you on lipids. Remember, our 4 types of lipids are fats or oils, phospholipids, steroids, and waxes. I have a few questions for you before we go. Have a great day, guys, though. Stay healthy. Hold on, I gotta go wax my car.